Okay, so now we're going to continue with a discussion on the cell and how cells divide and reproduce. And this includes the process of mitosis, where you're just splitting one cell, making a copy of that cell, and then having two copies of that cell, or meiosis, which is the production of gametes or sex cells. But first we're going to talk about a more fundamental way of splitting, and that's the way bacteria divide. It's similar to mitosis, but also has some significant differences. First, they don't have multiple linear chromosomes. Instead, they have just one circular chromosome. So the first step is that single circular bacterial chromosome is replicated, and it's done that by identifying the origin of replication and then going in both directions until a complete chromosome or two complete chromosomes are left. And those then end at the site of termination and separate. So now you have two chromosomes. Those new chromosomes go to opposite ends of the cell, and then the septum forms in a process called septation, and that bacteria then splits into two daughter cells, which are exact copies of each other as far as the chromosome and the genes within it. Eukaryotic chromosomes are different, of course, because they are linear. Every species either has a different number or a different arrangement of genes within those chromosomes. Humans, for example, have 46 chromosomes, 23 almost identical pairs, the exception of that being the X and Y chromosome, which are different in size and code for different things as well. Additional or missing a chromosome is usually fatal and ends in termination of a fetus. Chromosomes are made of chromatin, which is a complex combination or interaction between DNA and protein. Heterochromatin is the chromatin which is not expressed and is sometimes called junk DNA, although it does appear to have functions and helps the total functionality of the genes and their expression. Euchromatin then, or true chromatin, is what is actually expressed as a gene, or what makes proteins that determine your hair color or the thickness of your skin, or so on and so forth. DNA of a single chromosome is one long, continuous, double-stranded fiber. The typical human chromosome is 140 million nucleotides long, which is, if you unraveled it, would stretch from here to the moon. The proteins that interact with the chromosomes, some of them are called histone proteins, and what they do is they attract to the DNA itself and form bonds and make these coiling structures so that you don't have a strung out piece. So they are attracted to the negatively charged phosphate groups, and they are positively charged, so they create those bonds there. These are then wrapped into higher order organizations or coils called solenoids, and this allows for maximum compaction. So the DNA wraps around the histones. Those nucleosomes then also interact together, and they form the higher order solenoids. A karyotype then is a particular array of chromosome. So this is a karyotype of a human here. Humans are diploid, meaning they have two sets of chromosomes of the different kinds. One set they get from their mother and one set from their father, a total of 46 chromosomes. A haploid organism or cell has only one set of chromosomes and is represented by an N, where diploid would be 2N. A haploid human cell has 23 chromosomes, and you would see that in an egg or a sperm cell. Each pair of chromosomes are called homologous chromosomes. So chromosome 12 here, this is one of the homologs, and this is the other of the homologs, and these are homologous pairs of chromosomes, a homologous pair of chromosomes. Okay, replication is a much more complicated process than in the prokaryotes. Before replication can occur, each chromosome is just composed of one DNA molecule, or one copy of all the genes. After replication, each chromosome then has two copies. So this is still one chromosome, but it just has an extra set of DNA. This is called a chromatid. Then this chromosome has two chromatids. This chromosome only has one. They are held together by what are called cohesion proteins, and these are visible as two strands held together at a center called the centromere. One chromosome composed of two sister chromatids then is called a tetrad. So this is a tetrad and this is a tetrad. These are homologous chromosomes, as are these, but these chromosomes only have one copy and this has two copies. Okay, so each cell goes through a series of stages in which different functions occur, and that's called the cell cycle. Interphase is where most cells spend most of their life. The first phase is called G1, or gap phase 1. The second one is synthesis, and this is where DNA replicates, or the S phase. And then the third stage is G2, or gap phase 2. This is where organelles replicate and the cell itself is getting ready to divide or make another copy of chromosomes. And that happens then in the M phase, or the mitotic phase. The first step in mitotic phase is mitosis, which is then subdivided into 5 phases, which we'll talk about. And then cytokinesis is the actual separation of the two new cells. All right, so interphase. Interphase is G1S and G2 phase, like we just talked about. And in this phase, the chromosome looks hard to distinguish anything within it, because the DNA is being unraveled and used to make, the genes are open and being used to make proteins. And so the nucleus itself just looks like a dark ball. The chromosomes themselves have this point of constriction after the S phase. And then the kinetochore is the site for microtubules to attach. So if you remember, centrosomes, what they are for is for guiding chromosomes during mitosis. One of the things they do is they have microtubules which come out and attach to the chromosomes. And the chromatids then stay attached by the centromere at the cohesion molecules, or the cohesion spot there. Mitosis then is the process of splitting these chromosomes and splitting them so each homologous chromosome, or each chromatid on a chromosome, splits and goes to opposite ends. The first phase is called prophase. This is kind of the preparatory phase. Then you have prometaphase, where the spindle apparatus starts attaching. You have metaphase, anaphase, and telophase. We'll actually go through these in more detail. So the first one, prophase. Chromosomes condense and are then ready to attach. The spindle apparatus assembles, and this is what's going to then attach to the chromosomes. They then move to the opposite ends of the cell and form what are called asters, or a radial array of microtubules. And then the nuclear envelope breaks down so that those connections can begin to form. In prometaphase, that occurs after there is no more nuclear envelope, so it's completely dissolved. Then the microtubules attach, and they attach to the kinetochores, and each sister chromatid then is connected to an opposite microtubule. In congression, then, the chromosomes begin to move to the center of the cell, as the motor proteins at the kinetochores then align them. Metaphase is where they meet then across the middle, and there's no like physical place where the metaphase plate aligns, but it's more of a like just an imaginary line where they all line up, but it's generally in the middle. This is also the future axis of where the cell will divide, but there's nothing physically there denoting the place. In anaphase, they separate, so they go to different ends of the cell. And there's two forms of movements. Anaphase A, the kinetochores are being pulled, and also the cell itself is elongating and pulling further apart. So that's anaphase A and B. In telophase, then, the spindle apparatus disassembles. So there's no more connection because we already have split the chromosomes. And the nuclear envelope then starts to form around those different chromatids, or chromosomes made of one chromatid. These chromosomes then begin to uncoil, and the nucleolus then begins to appear where those genes start to be used to make RNA. Lastly, we have cytokinesis, which is the splitting of the cell. In an animal cell, you get a cleavage furrow, and that is, you can see that here. It's formed through actin proteins. In plant cells, they have a cell plate which it forms, because not only do they have to split the cells, they also have to grow more cell wall. Fungi and other protists, the nuclear membrane does not dissolve. Instead, it occurs within the nucleus and splits with cytokinesis. All right, so that's mitosis. Meiosis is very similar. We're going to talk a little bit about the life cycle, sexual life cycle, and then relate that to meiosis. So the sexual life cycle, the endpoints of each are meiosis and fertilization. In most organisms, you have a diploid cell, which are the somatic cells. So these are the cells of the body. And then you have haploid cells, which are the gametes, which are the sperm and the egg. And these have only one set of chromosomes, while the diploid cells have two. The offspring then inherit genetic material from both parents, one haploid cell from each. These then fuse to become diploid. Life cycles then show this alternation of generations, or alternation between a haploid stage and a diploid stage. In most animals, the diploid stage is dominant, but in some animals have haploid stages as well. In humans, the haploid stage is very small, very short. But meiosis then occurs in sexually mature adults to form sperm and egg. Meiosis is also called reduction division because you are reducing the number of chromosomes in each cell. So the daughter cells have one homologue from each chromosome pair. It's very similar to mitosis, but it has two rounds of division. Meiosis 1 and Meiosis 2, and each of them have a prophase, metaphase, anaphase, and telophase stage. There's no DNA replication between the meiotic division, so it doesn't double, and that allows it to split. When it splits twice, it reduces the number of chromosomes. And then the sisters are separated for each homologue, the sister chromatids. All right, so meiosis then has two phases, meiosis 1 and meiosis 2, and we'll go through each of those. One of the events which is very important for meiosis in allowing genetic recombination, or allowing a diversity of different types of genes to be expressed in the gametes, is the process of crossing over. And what happens in crossing over is genes, or the tip of one chromosome, will exchange with its sister chromosome. And so if you got one from your mom and one from your dad, those would come together, and part of the mother's gene would go on the dad's chromosome and vice versa. This occurs in when the chromosomes come together in a process called synapsis, and the site is called the chiasma site, which means cross. Okay, so in prophase 1 then is chromosomes coil tighter become visible. The nuclear envelope disappears. So it's very similar to prophase in mitosis. Each chromosome at this point is composed of 2 sister chromatodes because it went through replication in S phase of interphase. And then synapsis occurs. And during synapsis, those homologous chromosomes come together and are closely associated. And they remain attached at these chiasmata. These then move to the end of the chromosome arm before metaphase 1. In metaphase 1, the terminal chiasmata hold the homologs together following crossing over, and they go to different line up, not all the mothers on one side and the fathers on the other, but just randomly in the middle. Microtubules from opposite poles then attach to each homolog, and they are not at each sister chromatid, which is what happened in mitosis. And so these homologs are aligned at the metaphase plate side by side. So in mitosis, they were basically right on top of each other, but now they are next to each other. So when you pull them apart, it actually gives you a genetic diversification of different types. And this is called independent assortment. So they align at random at the metaphase plate, and they are separated after crossing over. And all these things allow for genetic recombination to occur. At anaphase, again, they separate, go to the chiasmata break, they go to different parts of the poles of the cell. The sister chromatids remain attached at their centromeres, and then each pole at this point has a haploid set of chromosomes. And they are independently assorted. In telophase 1, then the nuclear envelope reforms around each, and then cytokinesis occurs as the cleavage furrow separates the two. They are no longer identical. Now you have 2 sides of the cell which are different. And then meiosis 2 will occur after some interval of length. In meiosis 2, this is a lot like actually mitosis, where the chromosomes align on top of each other, and then they separate the sister chromatids. So they are pulled to different parts of the cell. And now you have 2 haploid cells at the end, each of them with a different set of chromosomes. So the result is you have 4 cells that are haploid, and these then develop into gametes such as an egg or a sperm. In plants and fungi and other ones, they may divide at this point mitotically, so they may make copies of each other and grow that way. And this produces a greater number of gametes and varies the number of gametes. Sometimes errors occur in meiosis, and this is called non-disjunction, where one of those chromosomes doesn't go the way it's supposed to, leaving the gametes with one too many or not enough chromosomes, and this can lead to termination or can also lead to some disorders such as Down syndrome. Alright, so just some differences between the two. Synapsis and crossing over only occur in meiosis. Sister chromatids remain joined at their centromeres throughout meiosis 1. Kinetochores, or sister chromatids, attached to the same pole in meiosis 1. And DNA replication is suppressed between meiosis 1 and meiosis 2. So these are characteristics which are unique to meiosis. And we'll talk about more of them in class. Okay, so just briefly, in meiosis 1, in prophase, you have the nuclear envelope disassembling. You have homologous chromosomes lining up, going through synapsis and crossing over. They then line up on the metaphasal plate, and rather than lining up as chromosomes, the sister chromosomes line up together, and then they are separated so that each new cell has 2 sister chromatids. So they still are aligned in their tetrads. And then in meiosis 2 is when they line up and the sister chromatids actually separate. But they've already gone through random assortment and crossing over, so now you have 4 haploid cells, and each of them has a unique combination of genes. And that's it.